Yeah, so thank you very much for inviting me to talk here today. So yes, I've been working at DeepMind for about 4 years, most of that time on the protein structure prediction problem. And with a large group of collaborators, the actual core team is really the first 5 or 6 names here, but we've had lots of input from other people who worked in other areas of DeepMind and provide a lot of ideas and support to this work, and including David Jones, who is a professor at University College London and now at the Crick Institute, which is about a block away from the DeepMind office, and he helped to set us up with telling us what the problem really is in protein structure prediction, and telling us how to, what data we should be using and what things we needed to worry about, and keeping our train and test sets separate. So he gave us a lot of the domain knowledge, whereas the original core team had no protein domain knowledge at all, though we did, during the course of this work, hire a couple of people who have some background in protein. So Chong-Li Chin and John Jumper both have PhDs from that relate to protein structures. So DeepMind is part of Google. It was a startup that was acquired, and DeepMind's core mission is to solve artificial general intelligence and to use it to solve important problems. Initially the focus was on the artificial general intelligence, and the core of the research team at DeepMind is sort of incrementally working towards this artificial general intelligence, which is not just solving specific problems like speech recognition or object recognition, but having a general intelligence that can do sort of the full range of tasks that we expect of human intelligence. And a lot of their work has been based around solving games as sort of stepping stones to achieve, to get towards AGI. And there's a roadmap there of sort of building up successive skills to get to what the components that we think will be necessary for AGI, of memory, imagination, planning, those sorts of things, as well as the sort of more solved problems of object recognition and speech and so on. We've had this mission of using AGI to solve important problems. So for a long time we've had an applied group that's, since the acquisition by Google, has been working on solving Google's problems, so recommendations in YouTube, braking in self-driving cars, these kind of things. But in the last few years, we've increasingly focused on science problems, and we now have a science team that sort of crystallized around the protein folding team, and our sort of scope is to apply these techniques to anywhere in science where we think we can make fundamental changes by applying the techniques of AGI and machine learning. But protein folding actually started out as a hackathon project that somebody just did a proof of principle, and then it's grown into a much bigger project as we found it was a very good application area for us. So protein folding is the problem of predicting the structure of a protein given the sequence of amino acids that form the molecule. So basically we're given essentially the chemical formula of the protein, and we have to predict its 3D structure. And this is a video of our system for generating the 3D structure. It's a temporal system that does it. So in this case, it sort of relaxes into the structure that it believes this protein should have. So that's what we're trying to do, is generate these structures. And it's a great application area for us because it's a very fundamental science problem. But it has a very clear goal of getting the structure, which is measurable, and it has rich data. There's a lot of data that is available to apply computer-based techniques to. And if we can make progress in this, then it will have impact in a lot of areas of biology for understanding protein interactions, the function of those proteins, which are essentially the machines that carry out nearly every function in living organisms, and will understand the processes of life, and it will have sort of implications in drug discovery, disease understanding, and so on, although we're trying to solve the fundamental problem. So the problem that we're solving is we get the amino acid sequence of which we encode as a string. Basically, there are 20 types of amino acids, so we can give each one a unique letter. And they form a one-dimensional chain. So we can express a protein as a sequence of amino acids, and we want to predict the three-dimensional structure of that. And that's essentially the curve that is followed by the backbone of this molecule, and then the actual individual types of amino acids give these side chains, which are highlighted in green here. Those are the individual differentiating characteristics of these individual amino acids, but the backbone is a common piece. Every amino acid has this sequence of nitrogen, carbon, carbon atoms. A group of those three atoms define the backbone, with the side chain distinguishing each amino acids. And we want to, we've basically focused on the problem of working out the backbone geometry, not so much worrying about the side chain geometry, but the backbone geometry describes the overall shape of a protein. One way of representing this is through torsion angles. The geometry of this backbone is highly constrained. The bond lengths and angles are set by the physics of the molecule. So you can essentially characterize the backbone geometry by a set of torsion angles, this phi and psi. There's another one, omega, which is nearly always 180 degrees. So in fact, in our modeling, we just always left it to be 180 degrees. And so we can see this as determining values of phi and psi. That's a nice, simple representation of this geometry. So if we have a length L protein here, then there will be two L parameters, two L phi parameters and L psi parameters. And one fundamental piece of our system is that we wrote a TensorFlow model of protein geometry, that if we give it a set of phi's and psi's, then it will give us the coordinates x out. And that's a fully differentiable model, so we can differentiate through that model to optimize these phi and psi angles, and we'll come back to that later. So we focused as a way of assessing whether we were working on this problem, whether we were achieving any progress. We decided to focus on this CASP assessment. It's the critical assessment of protein structure prediction, something that David's been involved in for many years and been very successful at. And it's a blind structure prediction competition. So every 2 years, a set of newly solved structures, they're being solved by experimental techniques like X-ray crystallography, nuclear magnetic resonance, or more recently, cryo-electron microscopy, to solve the real structures. To some extent, those structures are dependent on the experimental techniques, but we treat them as the truth for our purposes. So these newly solved structures are about to be published in the public data set, the Protein Data Bank. They're withheld for the purposes of this competition. The sequences are released to competitors, and then we get up to 3 weeks to predict the structure and return it to the assessors. So it's a blind prediction. Nobody has seen these structures except the experimental group that solved the structure. And there are around 90 groups from labs all around the world, mostly university groups, who are entering their prediction methods, including the Rosetta from David Baker's lab. And then scoring is done after the fact, basically by looking at the structures and looking at the answers. The proteins are divided into hard and easy targets, and the scoring is done. It's divided up into these two categories of free modeling or template-based, where template-based modeling is it's still a structure prediction technique, but it uses solved structures much more heavily, where if you can find a protein with a very similar sequence to the one that you're being asked to predict, with a known structure, then you can infer that the structure of the one that you're trying to predict is going to be similar to the one that has a similar sequence. So you can base your prediction on that solved structure, which is called a template, and you just need to make adjustments to take into account the differences in sequence between your target protein and the already solved one. So that's a very well-established technique that gets much better results because it's based on these templates. And we deliberately didn't focus on that task. We designed our system to work on the free modeling task, where there isn't a specific structure with a very similar sequence. You're trying to infer generally from the sequence of amino acids what the structure might be. And in the dividing line between these two categories is rather blurred. With hindsight, when you've solved the structure, you can actually very often say, oh, well, there was a solved structure that was very similar. And if we'd found it, then we might have got a much better answer. But finding that template in the first place is actually quite a difficult task in some cases. So that's partly why the assessors do this after the fact. They're dividing these targets into the free modeling and template-based. Another thing is proteins fold into domains. You may have one large molecule that essentially folds up into separate pieces. And those pieces can get spliced in the genome, so they're brought together. You may have a small protein that folds into a ball that is joined in the genome to another one, and then those actually end up forming one single molecule, but essentially the two pieces may fold almost independently. So people divide proteins into domains, and to a large extent, people consider those as folding separately. So if you can split a protein into two domains, you can fold the pieces and then stick them together after the fact. That's a simplification that people apply, and it's also used for scoring. So the scoring of CASP is based on domains. So I'll go straight to the results. This is the results of the CASP assessment in the free modeling category. This is a combination of free modeling and the sort of borderline category where they're sort of hard free modeling, difficult template-based modeling categories. Using a scoring, so it's measuring how close your structure is to the ground truth, using these metrics, which again, CASP has this unusual system whereby the assessors choose the scoring metric after the competition has happened, although it tends to be quite similar from round to round, but they sort of reserve the right to change it. So this year they combined GDTTS, which is the sort of well-established metric that really measures the geometry, with a quality control score that measures other aspects of the protein, how protein-like it is in other ways. So we were sort of trying to maximize this GDTTS score. In fact, we hadn't even heard of the quality control score until the CASP assessment. But these are the results. There's quite a complicated method of using z-scores to normalize the results, but then they accumulate the z-scores across all the targets. I think it's 43 targets for this FM. And you can see that our system, AlphaFold, had quite a lead over the nearest competitors in this free modeling. And even though we weren't doing template-based modeling explicitly, our system generalized to template-based modeling quite well. If you use this particular metric, then we actually came out slightly ahead. You're allowed to submit 5 results to the structure predictions, and the assessors either rank the one you designate as your best one, or they rank all, they take all 5 and they take the best of those 5. If you use the best of 5, then we're slightly ahead. If you use our top 1, then we're second or third, I think. So surprisingly, even though we didn't do a template-based modeling system, our system did actually generalize quite well to templates. And the scoring here is a much more complicated formula that the assessors use for these generally higher quality predictions. So this is an overview of our system, which now I'm going into a little bit more detail of the sort of protein-specific aspects. So a standard technique for protein structure prediction is to find similar sequences. We have very large data sets of protein sequences that we know exist, even though we don't know the structures. And we can infer commonality between those sequences. If we can find a list of similar sequences, we can infer that they probably have the same structure, because the chance of two protein sequences looking similar by chance is very small, because protein space is very large. It's 20 to the n possible proteins for a protein of length n. Very few of those are actually realizable or stable and useful. So nature conserves useful proteins, and so nature has explored a very small space of protein, the possible proteins. So if you find similar proteins, then you can infer that they came from a common ancestor, and they probably have a very similar structure. So a very well-established technique is to look for similar sequences in these large sequence databases, and you can use co-evolutionary features to try and find correlations in those, which I'm not going to go into in detail, but they can provide some indication of where there are contacts within a protein, where the protein folds back on itself and two residues come into contact. Then you'll get these contacts implied by conservation or covariation of two residues. So we use a standard feature extraction method there to extract features from these similar sequences. So we may get it on the order of 1,000 similar sequences. And the number of similar sequences that you find with this HH-Blitz tool in the Uniclust data set is a very good indication of how good the predictions are going to be. If you find lots of similar proteins, you've got a lot of information for predicting the structure. If you don't find any, then it becomes a lot harder. We then trained a deep residual network, a neural network, to predict histograms of distances, and I'll go into more details on that. We predict other attributes of the protein. Basically, we're predicting features of the protein. And then we try and realize a protein structure that obeys the predictions that we make with the neural network. And as part of this, we are also using Rosetta, the tool from David Baker's group, to get some additional measures of the quality of the protein, in a sense. So the main feature of our system is we make a very large number of predictions from this neural network of the distance between pairs of residues. So we number the residues of a length n protein as with the index I or j, and we want to know the distance between a pair of residues ij. We're not going to know exactly what that distance is, so we predict a probability distribution, like a histogram of what those distances could be. And we also predict what those torsion angles are likely to be. We can get a prediction of the distributions for those torsion angles. And for our system, it fundamentally relies on these distance predictions. We get a small benefit from predicting these torsion angles. But one thing about using a neural network to predict these is that they're calibrated. So if we predict that there's a 10% chance of the distance being between 3 and 4 angstrom, or 8 and 10 angstrom, then on average there will be that percentage of those predictions will have that value. So they're calibrated, they're consistent. They're smooth. We find we get smooth distributions, and they're very detailed. That we get this fine resolution. We decided in retrospect our predictions were actually too fine resolution. We get slightly better results with lower resolution, but they are very detailed. Previously, people have focused on predicting just contacts. Is the distance less than 8 angstrom? And what we and a couple of other people in this CASP competition did is we went from this contact predictions to distance predictions of saying not just is it less than 8 angstrom, but what value will it take basically dividing, instead of having 2 bins in the histogram, we went to many more. And that gives us a lot more detailed information. The covariation is this signal that tells you whether residues are likely to be in contact. But you can get a lot more prediction than just are they going to be in contact. You can actually start predicting the distances. And one thing is that, because we make many of these predictions, we're basically making n squared of these distance predictions, we're getting lots and lots of pieces of information about the structure. And the match between a given structure and these predictions gives us a scoring function. We can rank 2 different structure predictions by using how well they match these distances. Are the distances likely under these predictions? So we can actually measure the log likelihood of a given structure under all of these distributions. So I'll just very quickly go over deep learning. So basically, deep learning is training deep neural networks with multiple layers. Basically, after the renaissance of neural networks, one of the critical things was that they just became a lot deeper. They became a lot bigger than they had been in the 90s. So deep learning is the sort of tag that stuck for the difference. But really, a lot of that is just about computational power. We have the computational power to train bigger, deeper networks. And basically, they're function approximators where you're trying to predict some value. In our case, we're trying to predict these histogram probabilities. And you can train them to optimize an objective. In our case, the objective is the log likelihood of all the structures in our training set. We're trying to maximize the likelihood for all those structures. And we do it by changing the parameters of the neural network, which are the weights inside the network that are used to parameterize this function. And we can train it by gradient descent, by taking incremental steps in those parameters that each incremental step is designed to increase this objective by showing it training examples, tweaking the parameters that increase the likelihood of the current training examples for our case. And you've seen it's been hugely successful in recent years, revolutionizing many domains. So I worked in speech recognition and speech synthesis. Those are completely neural network based now and working at very large scale in things like the Google Assistant. Machine translation, again, Google Translate is all based on neural networks now. Many, many different image tasks like Google Photos. And then what DeepMind has sort of focused on is agents, where you're training something to actually act rather than just making a prediction or a classification. And we focused on these games as sort of controlled environments that you can simulate very well in software. And we've had a series of papers with applying neural networks to playing Go, chess, Atari games, and more recently StarCraft. But people are also applying these neural networks to things like self-driving cars and sort of any domain you can think of, really. And for our purposes, we're interested in them. They're capable of modeling this very complex data. It's very hard to understand the mapping between the protein structure, the protein sequence, and the structure. But we have a lot of data that's available. And there are these very long-range, subtle patterns where residues that are far apart in the sequence actually come physically close together. So you have these long-range interactions. Protein sequence space is vast, so we can't possibly model every single sequence. We have to generalize across repeated patterns and motifs that we find in the training data. And neural networks are very good at extracting the commonality in those different across the training data and generalizing to new sequences which are similar in some respect but have never been seen before. But the thing with neural networks is you've got to structure the network to give what's called an inductive bias so that the structure of the network is mapped to the problem that you're trying to solve. You can, originally all neural networks were sort of fully connected direct networks where you feed in all your features at the beginning and you get your output out at the end. But if you structure your network, like having a convolutional network for images, that's the sort of classic example that's been around for longest, then you get an inductive bias to model specific features of the task that you're interested in. So for images, you want, in most cases, you want translation independence, that if I'm classifying a dog in the bottom left-hand corner of my image, it's going to look exactly like a dog in the top right-hand corner of my image. So I want to apply the same operations equally to different parts of an image. So I can structure my network so that it has that invariance. And so you want to design your network to have the invariances of your data. So for protein structure prediction, it's a very complex problem. It's very hard to understand really what's going on in detail, modeling these complex long-range interactions. But fortunately, there's a large amount of data. There's this protein data bank, which has of the order of 150,000 solved structures. Unfortunately, that's highly redundant. So it's not the scale of many of the problems that that deep learning has been applied to before, where you have millions of utterances for speech or millions of labels of images. But nevertheless, it is quite rich in that we have full structures. We have these sequence structure pairs. In practice, we remove the redundancy and have a much reduced training set. And then the CASP assessment provides us with a benchmark with very well-defined goals. So we train a network to predict these inter-residue distances. We're extending previous work that's in recent years has transformed the field by predicting contacts, which give us these long-range interactions. So what I have here is a distance map. So if I label my residues I and J, this is the distance between residues I and J. So bright is short distance. So the diagonal is all zeros. It's the distance between a residue and itself. But off diagonal, you get small distances. In this particular region is the helix. That basically, as it wraps around, residues come close to their near neighbors. So you get this bright band near the diagonal. And then you get these beta sheets where two pieces line up. And then you get these long-range things which are far from the diagonal. So they're residues which are distant along the sequence, but are actually close in space. So you've got these bright pixels here. And so we want to predict these distances. Previously, people have predicted the binarized version of this, of just identify the bright spots. But we can see that there's a lot of structure beyond just is there a contact or not. And in particular, if we look at the interaction between this helix and this strand here, we get, in that interaction, we get a few bright spots with a periodic structure that reflect the nature of this helix and this strand. And we're getting, in what is considered a longer range independence with dependency, we're off the diagonal. These two structures are telling us a lot about the pattern, not just of the contacts, but the individual distances here. As the bright and dark variations here are respecting this geometry. If we know that we have a helix here and we have a strand here, there are only a limited number of ways that they can come together, and so we have a lot of constraints by those two pieces on how those interact. And the full distance matrix in this box between these two pieces tells us a lot about those possible distances. So we're not going to predict exact distances, we're predicting distributions, but we've highly constrained it by knowing what these secondary structure pieces are. But we may also have some covariation that tells us, oh, this particular spot is a contact, because we see in these similar sequences the covariation. So we may nail down that these two pieces are in contact, and then that helps us, knowing the secondary structure and those contacts helps us limit the values of those possible distances. So the network has a lot of information to go on to make those predictions. So we trained a very large two-dimensional network. So it takes an n by n region. In our case, we take 64 by 64 patches and try and predict all the distances in that region. So we're feeding in the sequence for a piece of length 64 and another piece of length 64. So those correspond to, say, this region here and this region here, if they were 64 long. And then we're trying to predict all the distances in a 64 by 64 square, which isn't necessarily on the diagonal. And we do this by a softmax probability distribution, which is well calibrated. So it's going to give us a probability distribution like this, with these distance bins. We chose originally 40 half angstrom bins. We later went to 64 bins, although more recently we've decided that it doesn't need that full resolution, and we don't need to go out to such long distances. And we call these distance histograms or distograms. And basically, we're predicting the marginal probability distributions for each ij pair. So these are going to be highly correlated. We're not predicting the full joint distribution of those distances. And we're basically doing this for an n by n region. So it's very similar to a lot of the standard image-based networks that take an image and make some predictions, like a segmentation of an image. And so we use this residual convolutional network, which is a well-established architecture. The data that we use is based on this protein data bank, which gives us the structures for a set of known, a set of sequences. We simplify this by splitting it into these separate domains, so these pieces which we think fold largely independently. We deduplicate those using the Cath dataset, which clusters very similar things together, and we take one example from each cluster. So we end up with 29,000 different cluster centers, and we just train on the cluster representatives. And then we also use the Uniclust sequence database to find these multiple sequence alignment-based features that tell us about the covariation. And we use HH-Blitz and SciBlast to find these similar sequences, and did our own implementation of these covariation features, but it's essentially the same as a standard tool called CCMPred. And we didn't use mutual information for our particular network, but that's another standard feature. So this is an example of the Frobenius norm of these 2D features that we use. That, this can give you information about covariation for a particular spot that these residues are in contact. We also have these 1D features, which are called the profile and the original amino acid sequence. And then for a given 2D location, we replicate for I and J, we reproduce the sequence-based features for I and for J. These represented by these vertical and diagonal lines, and we have the 2D information for the position ij. And we just feed that into this sort of standard network architecture. This was developed by Laurent Siffre, who actually worked on some of the Go work. So he optimized this architecture originally for contact prediction. And it's a set of standard building blocks that are used in neural networks now. So this is a residual network. We work at with 128 features, but then within each block we project down to 64 features. We use an LU nonlinearity, and we use this batch norm to keep numbers within range to improve training speed. And then do 3x3 convolutions inside these residual blocks. So dilated convolutions allow you to have incorporate features from not just the neighbors of a particular pixel, but from further and further afield, but without increasing the computational complexity, because at each stage you're only looking at 9 possible pixels, but they're just further and further apart. The dilations allow you to look at a bigger receptive field. And then we stack these residual blocks, and in the end, the network we have is ridiculously deep. It's 220 of these residual blocks. That was the architecture. We kept on making it deeper up to this point, and it kept on getting better. But the network has 21 million parameters, which is a sort of moderate amount of parameters. Compared to some of the networks, which now people have networks with billions of parameters. And we just feed it through this network, and we end up with these n by n distance predictions, each of which is, say, 64 bins. And there's some previous work which had applied residual network, or some concurrent work that has already been published now on using these deep residual networks for contact prediction. And we use, in some cases, a similar architecture. We got to a slightly different thing, so they used slightly different embeddings. But one critical difference, we think, is that we train not on entire proteins or entire domains, but we crop out 64 by 64 regions. So when we get a given example, let's say this is a whole domain, we chop it up into these 64 by 64 regions, and we train the network just to predict one of those regions at a time. So this improves efficiency because the network always sees 64 by 64. It means we don't have trouble scaling to very large proteins, and we don't have inconsistencies between short proteins and large proteins. So it's very efficient in terms of speed. But it also has benefits in that every time you see a protein, you split it up into many, many training examples. So just one cropping, tiling of this into 64 by 64 regions will give you, in this case, 16 training examples. But the next time we can choose a random offset, and we'll get a completely different set of crops. So from our 30,000 training examples, we end up with millions or billions of possible training examples. And going through the data over and over again, we never see exactly the same training example. And that gives data augmentation, which is a very valuable attribute for neural network training. A lot of people have done a lot of work in devising clever ways of doing data augmentation. So I know other people in CASP tried different data augmentation techniques. We also applied dropout, which is another technique, which is a regularization technique, which is similar to, has similar attributes to data augmentation in that you basically effectively add noise to your inputs, so you get slightly different examples each time, although you believe the labels should be the same. And at test time, we can also try different crops. We don't have, we still put the crops through and we average them together. So we get averaging of all the different versions of the tiling of a given protein. And in practice, we actually trained 4 different models and averaged those together. And doing all those combinations improved our accuracy. So this is an example. This is a small target again. This is the true distance map. This is our prediction. So we're quite good close to the diagonal, where there are a lot of constraints about the chain geometry. We managed to predict these off-diagonal where the strands line up. And then we're getting some information here about the interaction between this strand and this helix. And remember, this is actually just the mean of the distogram. So there's actually a lot more information. If we look at this particular residue here, these distances, so here I've highlighted all the distances that are actually less than 8 angstroms. And here, these are all the distances from that particular residue. The actual distributions, and in red, I've drawn the true value. In gray, this is the threshold between 8 angstroms, which is the usual distinction between a contact and a non-contact. So the green distributions here are the ones that should be true contacts. So the red line is always to the left of the gray line in the green distributions. And you can see that the network is doing a pretty good job of predicting these distributions. That in the green examples, the peak is pretty nearly always left of the gray line. We get a pretty high value at the true value, the red bar. And then for the blue distributions, then generally we're making larger predictions that the probability mass is off to the right. So we're getting a lot of information about what those distances should be. And in a lot of cases, even when the distance is large, we're predicting exactly what the distance should be. The peak of this distribution here is at the true value. So if we can get the network to make structures that have the distances that we're predicting, then we should be getting pretty good structures. Okay. One thing is we measured the performance of just predicting the distances, how good the contact prediction is. That's a standard metric. People haven't been using metrics to predict distances because this is really the first time people have been really using distances. But if we sum the probability mass below 8 angstroms, we can get a contact prediction accuracy. And this is some examples on a previous data set. For a single model, we were getting a 50.7% precision for long-range contacts. When we ensembled 4 models, we go up to 52.3%, so that ensembling is helping us. Without the multiple sequence alignment features, then we go down to 13.6%. So we're heavily relying on this covariation from similar sequences. And then I'll come back to later, we also have a reference model that doesn't know what the sequence is, and that gets very poor accuracy, but I'll come back to that. These are the actual sort of metrics that are compared usually. We didn't enter the contact prediction category of CASP, but the 2 best competitors that were evaluated for that, we compared our accuracy for contact prediction, and we're getting better scores for the long-range contacts in every category, whether it's for free modeling, the template-based modeling / FM, the sort of intermediate category, or the pure template-based modeling. We're getting much more accurate, 3% here, 6% here, and that extra accuracy translates into the ability to realize better structure. And this is just in contact prediction, but in fact we have this much richer information of distance prediction. In fact, Raptor X also was predicting distances, but we were able to use those distances to constrain our model in a richer way than the other structure prediction methods that did use distance predictions in this CASP. We also trained the network to predict some other things. So we tried to do secondary structure prediction of is it a sheet or a helix. We think we're getting around state-of-the-art predictions for secondary structure prediction because essentially the network knows, is learning whether it's a helix because it's got to do this broad diagonal, or a sheet. It will have a particular structure and we will have these extra diagonals. So the network knows what the secondary structure is. So we had an auxiliary loss on the network. Auxiliary losses have been found to help neural network training in a lot of cases. In a lot of our experiments, it did help to predict secondary structure, but for our final system, I think we decided that secondary structure prediction as an auxiliary loss didn't actually improve our performance for contact prediction. We also trained the same network to predict these torsion angles. So these are what are known as Ramachandran plots. So for a given residue, this is residue number 19, it's predicting the probability distribution of phi and psi. And we wanted this additional constraint of what these torsion angles could be to initialize a model with some starting values of torsion angles. So in some cases, it's giving us very highly constrained values of, somewhere in this, it's very likely to be somewhere in this region. And maybe you can see there's a small island of probability there. We're highly constraining the possible values of torsion angles. Another thing we did in the system is, rather than just predicting the probabilities are highly constrained, the distances are highly constrained by the geometry of the chain and by the nature of proteins. So there's a background distribution, and we really want to predict the sort of novelty for this particular protein, how it is different to the average protein. So we trained the network to predict the distances for an average protein. Basically, we trained the identical network, except we didn't give it any features that told it what the actual protein sequence was. So we told it basically the length of the protein by running these crops for a given length, but we trained it without any protein features except a thing that says is glycine, because that doesn't have C-beta distances. So the distances that are being predicted are slightly different. I won't explain for the non-protein people here. So we trained this network to basically predict what we would expect the distance to be for a given ij pair if we didn't know what the sequence was. And then we can subtract that probability distribution from the prediction that we have to give us the difference, which is the sort of what we learn by giving it the sequence. And this is a fairly standard method that's been used, for instance, in Rosetta. You predict a background distribution, and we use, so it's the difference in likelihood between the our prediction knowing the sequence and our prediction for the background. And in this case, we have these two peaks. But when we subtract out, so that the prediction is in blue and the background is in red, there's a high probability here in the prediction, but we would expect there to be a high probability at this point for this particular offset. So when we subtract out the background, we get this distribution, and we get, in this case, this is a negative log likelihood that we want to get. We want to find a minimum. We get these peaks, a trough here that we want to get into, and we only have a very small trough at this point. So we can add together all these scores, which are these differences, and we want to optimize that for a given. We want to find a structure that realizes that. We've got two methods. One is simulated annealing, which is a standard technique. So, for instance, Rosetta has used that for many years. We did that with a generative model of protein structure. But the new thing that we did here is to go from scratch with repeated gradient descent. So we initialize models of the protein with our guess at the angles, which are just randomly drawn from our predictions. And then we optimize by gradient descent these distance scores. So this is an example of a trajectory as we optimize those scores. We keep on doing it. We do it many times, initializing from random starting positions. This is one example. So just our guesses at the angles get some pieces correct. We get some of the helices, which are easy to predict. And then over time, as we do gradient descent, our scores improve. So the TM score goes up, the RMSD goes down, and we start to realize the secondary structure. Here we see the helices in red form. They're not present at the beginning in some of them. One of them is. And then we achieve these secondary structure elements as we go through an optimization. And if we do this many times and choose the final, across thousands of these attempts, we choose the one with the best score at the end, that's what we submit. I'll skip this. I think I'm running out of time. So this is an example of one domain where we did significantly better than the other competitors in CASP. And partly this is because for this method, we didn't chop it into separate pieces. This was how we guessed the protein should be chopped into separate domains. This is the true segmentation, and this is how we guessed it should be chopped up. And you see we've made a lot of mistakes. We guessed 3 domains. There are 3 domains, but in fact this one is made of 2 pieces. We got the answer wrong. But this gradient descent method, we were able to just optimize the whole chain in one go. So getting this segmentation wrong didn't hurt us as much as it would have done if we'd actually separated it. Well, in practice, we didn't do very well for this separated domain. GDT is well under 45. But we did significantly better both at predicting the contacts than other people and at solving the complete structure. So this is that full structure as it's optimizing step by step. Each of these steps is improving the score by a small amount. We're taking steps in the phi psi space through our differentiable model of protein geometry to get successively better scores. We just use LBFGS as the optimization algorithm in this case. And it gives us a structure which is compact and obeying as many of these distance constraints as possible. Here's another example where, in fact, our gradient descent method didn't do as well as our other method. The fragment assembly method actually did better. You can see the sheets coming out there. And this is just a measure of how long it took. So we parallelized this over many machines, but after 100 node hours, we're basically getting to the full GDT that we can get even if we run for much longer. That's per protein. This is but averaged over a large set of proteins. And then this just shows that our accuracy of structure prediction correlates quite well with our accuracy of predicting the contacts. It is dependent on the contact prediction. So overall, deep learning-based distance prediction gave us better contact prediction accuracy, but beyond that, it gives us a richer source of information. The actual distance information tells us a lot more about the structure of proteins than just knowing about is it a contact or not. Other groups, two other groups in CASP also moved to doing distance prediction this time in parallel, but we were able to use this rich information. They ended up thresholding the histograms, so they ended up with essentially a binary constraint on the distance, still having made these richer predictions, but we were able to use these smooth potentials that we constructed from these distributions, use the full distribution to realize our structures. Training with crops was very effective, even for modeling very long-range dependencies, very far off diagonal. We're still predicting the interaction of at an arbitrary distance separation. And we think the data augmentation was very valuable. Our final method ended up avoiding domain segmentation, and that was successful and probably helped us avoid some errors. We used Rosetta Relax from Rosetta tool from University of Washington. It gave us the side chain positions, which we didn't try to predict at all ourselves. It did slightly change the backbone geometry, but in the end, it didn't help the GDT, the score that we were trying to optimize. Basically, we were augmenting it with our distance prediction, so it was highly constrained to stay close to the solution that we'd already derived for the backbone. But it probably helped the quality control score by getting the angles and the side chains more consistent. And our methods surprisingly worked very well on templates, although it completely missed other templates. Say if there was a single template and very few other similar sequences, we were 40 GDT behind other competitors on some of the targets. So what are we trying to do now? We're still focused on the fundamental problem of protein structure prediction. We made a step forward, but we still got a long way to go. I think the GDTs here are, high 50s, low 60s, sort of average. We really want to get to 100. Nature can solve proteins even without the co-evolutionary information. We believe that ultimately we should be able to not use that information and still predict the structure. We're trying to publish a paper. We don't currently have a plan to put up a public server, but we expect that to come with time. And we're trying to learn what people will use our protein structure predictions for and what the accuracy requirements are. The structure, the quality got better, but it's not. Well, it's just getting to the point where we think that these predictions are relevant for biologists doing tasks. So, we're interested in finding out when people could use our structure predictions. There are some tasks where the general shape of a protein is enough, and there are others where you've got to get down to angstrom resolution, in the side chains, and we're far from doing that. So, we'll want to know when we'll be biologically relevant, or what things we're biologically relevant for already, and then we're looking at other things like protein-protein interaction, where two proteins dock together. Similarly, for small molecules binding to proteins, protein design, we've been using some of David's data for assessing protein stability for his designs. And then another area that we could get involved in is experimentally assisted structure prediction, where you use an experimental technique to give you some constraints on the structure, but then you apply your model to get even better answers than either could do by themselves. So thanks to many people, the CASP organizers and assessors, the experimentalists who produced the structures, many people who've provided tools, including the Rosetta tool that we used, and then we've used many other public domain tools. David Jones helped us get started, provided a lot of domain knowledge, and then this is the team who most directly contributed, but then we've had lots of input from many other people at DeepMind. So, thank you very much.
Thanks for the great talk. Questions for Andrew?
Thank you.
I'm a protein chemist. I'm a grad student. And I crystallize proteins. So the main way that people solve the structure of proteins is that they look at the sequence, they look at the side blast, and it's really interesting because I feel like when I look at sequence alignments, I can see the information content of different domains and motifs of the protein. But I've also been looking at what is really the physics of crystallography. And I think it's really interesting that the atomic structure of matter is really defined by crystals. But we don't know what are the rules that make a protein crystal. So they say that protein crystallography is an art form because you just have to kind of do certain steps and just empirically try to go towards the goal of making the protein crystal. And so I kind of question what is the validity of building a system on something that is going to define the laws of physics and kind of define how local order gives rise to global order.
I mean, ultimately, yes, we're very much exploiting the co-evolution information, and we're not modeling the physics. We do much worse if we don't have a multiple sequence alignment. We've tried training our system evo-free, as we call it, without the evolutionary information, and it does perform much worse. So we're heavily exploiting that. We're not yet learning very much about the physics at all. And we're not modeling atoms here. We are modeling residues as units. Yes, we're a long way from the physics, and ultimately we see that that's what we should be getting towards, that we should be learning a system that is modeling the physics, but hopefully doing it by choosing the right inductive bias of the model, and the hope is that this is a route to get there. The people are defining molecular dynamics force fields or so on, that's another approach, but hopefully we can get another route to that same answer, and we'll get closer and closer to the physics. But we're a long way from that, and we're sort of trying to make a tool that works in the short term for now. Yep. Yep.
Thanks for a great talk.
Thanks.
So by splitting each protein into these 64 by 64 crops, in my mind it feels like you're saying implicitly that those patches, or the distance between those residues, is going to be independent of any other information of the protein sequence. And I'm just curious if you tried different, I'm sure you did try a bunch of different sized windows or other types of ways of combining information. And did you find that 64 by 64 was like the optimal trade-off?
But to be honest, a lot of those experiments were done when we were doing contact prediction before we switched to distance prediction. That may not be the optimal, but basically we see that, if you've got two 64 by 64 windows, you're essentially modeling 128 residues. That is a lot of most domains, certainly, and we're only training at the domain level. So we think we've got pretty good coverage, and that we tell the network the separation between those two chunks. It does see the residue index as well. So we feel that it's got a lot of information to go on, and putting in the extra information, basically there was a trade-off versus this data augmentation and the efficiency. So maybe there is a way to exploit that full information better, but we think there's very little in it. And David Jones did a study in a paper where he changed the receptive field of the convolutional network, and he found, I think, 32 by 32 was sufficient for modeling those the long-range interactions. Yep. Right.
You have commented a couple of times that the multiple sequence alignment is very important, and yet you didn't use mutual information. Do you think that's redundant with the features you have used, or what's your thinking?
That's really just an empirical result of taking it out. Our results were as good. We had that feature in our data set. To be honest, I'm not sufficiently au fait with the details of what's, what those two features are modeling to know the contrast. I mean, we have a very rich representation of the covariation in that we don't just take the CCM pred single value per position, but we've got a 484 dimensional feature at each position because we've got the full POTS model parameters. So at each ij pair, we've got 484 dimensions. So maybe that's sufficiently rich to capture everything we need.
I think there's a question back there. Yeah, so optimization of deep learning is very forgiving in the sense that you over-parameterize everything, and then there's a lot of different subspaces you can walk down hill. But it seems that doing gradient descent on a torsion angle is much less forgiving, right? Because I have one torsion angle, and I switch it, and it's these huge two things not colliding. So I'm curious, have you guys tried other things besides gradient descent? Like, you can do all kinds of MCMC with gradient information, and blah, 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 blah.
So we did have this simulated annealing-based system where we did fragment insertion, which is very similar to the Rosetta fragment insertion, where we had a neural network end-to-end trained model of protein structure that generated torsion angles from sequence separate to the one that I've described here, and then we found that by itself it's not very good at predicting structures, but it was good enough at predicting fragments. So we would make predictions, chop them into fragments, and then do fragment insertion. And we also entered that into CASP. We we basically made predictions with both systems, and we ranked them. And so in some cases, that did better. But in the end, the gradient descent method was simpler and faster. So that's what we've emphasized. I think there's a slight edge in the results from the gradient descent method, but there's not much in it. It's basically the scoring function is the critical thing. We just found a nicer way to optimize, which in itself is not new. Rosetta has a gradient descent function, but the fact that we were able to go from scratch with gradient descent is sort of the interesting piece. Yeah.
Actually, what type of optimizer did you use there?
So that was just LBFGS. I think it was SciPy version of LBFGS. That was not the sort of critical thing, but the function to the differentiable function of protein geometry was important as well to be able to go from torsions to coordinates.
Right. Well, let's take
Yeah.
There are more
Okay.
questions that come down. People come down afterwards. I'm sure Andrew would be willing to answer questions. And remember, there's a lunch afterwards for those who are interested. Right. Let's thank Andrew again.